Well, out of respect for everyone's time, this is recorded, so I think we're going to go ahead and get started. Let me bring up just a couple of introductory slides here. There we go. Welcome, everyone. Want to thank you for joining us for this Nuts and More webinar series. This is a real treat here. We have Dr. Sean Mehlenbacher with Oregon State University talking to us about hazelnuts as a model for nut tree breeding. This was the keynote talk at the 2023 Northern Nut Growers Association Chestnut Growers of America joint conference held in Columbia, Missouri. What a great time and what a fantastic place. And in the gym, Dr. Mehlenbacher gave this presentation, but the audio wasn't the best. So he was very kind to record it for us. And so we have the opportunity here this evening to hear that in full. Just some basic housekeeping. Again, this will be recorded. We'll have this up on the Iowa Nut Growers Association website. Um, Speaking of that, this is where you can find those presentations from the 2024 Northern Nut Growers Association Chestnut Growers of America conference that took place in Syracuse, New York. And this is also where the Nuts and More webinars will be hosted as well. So check that out to share with your friends and colleagues. A little bit about the Northern Nut Growers Association, a nonprofit founded in 1910. The promotion of hardy nut-bearing trees and a few select minor fruits. So there's definitely some folks that are part of the nut growers for persimmons, pawpaws, things like that. Again, our membership is really broad and diverse, comprised of growers, nurserymen, breeders, just amateurists, you name it. We're an eclectic group, that's for sure. So wanted to point out the website, your gateway to the world of nuts, most definitely. And then just point out that we do have our 2025 annual conference coming up here. So be sure to get that on your calendars. We had a couple of initial planning meetings. It's going to be fantastic. We're going to be in Michigan, starting things off on Sunday, August 3rd, with Nash Nurseries and a tour of their facility. It's going to be a fantastic time. Great presentations, lots of chestnuts and other nuts. So join us in Michigan. We'll have the registration up soon, but get that on your calendars the 3rd through the 6th. Again, this is what it'll look like once we get registration up and going. So be there or be square, as they say. Well, without further ado, let's get to the reason you're all here. So this is going to be Dr. Sean Mehlenbacher, Hazelnuts as a model for nut tree breeding. We recorded this, so we have it dialed in as far as the time. And so give me 3 shakes of a lamb's tail, and we'll get that started. And then after the recording, Dr. Mehlenbacher is going to join for live Q&A. So be sure to either get those questions in the chat or be prepared to ask them here towards the end.
Good afternoon or good evening. This is a repeat performance of my keynote presentation at the Northern Nut Growers meeting in Columbia, Missouri, back in July of 2023. I'm Sean Mehlenbacher, the hazelnut breeder at Oregon State University. And the title of my presentation is Hazelnut as a Model for Tree Nut Breeding. So the first part of this is focused on the Oregon State University hazelnut breeding program. And then the second part expands that presentation to talk about what's applicable to other temperate tree nuts. World hazelnut acreage is expanding. We're in a good position. There is high demand for hazelnut kernels for chocolate products. Expansion in Oregon is possible because of EFB resistant cultivars from Oregon State University. EFB, Eastern filbert blight, a fungal disease caused by a fungus that's native to the eastern states of North America. We grow the European hazelnut in Oregon. Those new cultivars from OSU, Oregon State University, not the OSU or the other OSU. Those cultivars were increased rapidly by micropropagation. We went from 29,000 acres in 2009 to 94,000 acres at the time of the meeting in Missouri. Hazelnut acreage has been expanding in traditional areas. Those include Italy, Southwest France, Oregon, Georgia, Azerbaijan, and also the leading producer, Turkey. People have also been planting new orchards in countries where that have never before been important for hazelnut production. That is Chile, Serbia, Ukraine, Eastern Croatia, Western Romania, and so on. And many of these places have a climate that's less than ideal. So hazelnuts will be challenged in those new environments. Plantings in eastern North America face major challenges, twofold: a less than ideal climate, plus eastern filbert blight caused by a fungus that's on the native Corylus americana, the American hazelnut. And wherever people plant European cultivars, they are exposed to eastern filbert blight in eastern North America. Genetic diversity is essential for any breeding program. Our industry in Oregon was founded using introductions of Felix Gier, a Frenchman who imported trees from Bordeaux, France, to his nursery in Nevada City, California. Our leading cultivar, Barcelona, our leading pollinizer, Daviana, as well as Hall's Giant, Du Chile, and Montebello, all came from his introductions in the 1880s. Barcelona was the most important variety for many decades. In the late 1950s, our industry was interested in evaluating other cultivars to find something better than Barcelona. So Quentin Zielinski in the late 1950s imported more than 100 cultivars. And within the first 10 years, they had been through the collection and concluded that none of them was better than the Barcelona that they were already growing. So in 1969, Maxine Thompson started the OSU Hazelnut Breeding Program with funding from the Oregon Hazelnut Commission. Maxine retired, and I was hired in 1986, and I ran the program for 38 years. Genetic diversity. In hazelnut, we have no shortage of genetic diversity. And it doesn't matter how you look at that diversity, whether it be nuts, husks, or DNA markers. Corylus avellana, the European hazelnut, is incredibly diverse. In this picture, you can see differences in size and shape and husk length. Early in my tenure at OSU, I imported from Europe 447 cultivars from 26 countries, and more recently imported seeds and planted 9,288 seedlings from seeds collected in 14 countries. The biggest collections were in Turkey, Georgia, southern Russia, and Azerbaijan. Eastern filbert blight resistance is a major objective. Of the materials that we exposed to the fungus, we found that about 2% were resistant to EFB. That is, no cankers developed after inoculation. We continue to work jointly with Rutgers University and with others, and with Rutgers in particular, it's been a mutually beneficial arrangement. I also have built up a long list of international contacts, and they have been helpful to me in my career. The steps in a hazelnut breeding program, or I could start by the steps in any plant breeding program. First, you define the objectives. What traits do you need to constitute a better variety? And for each objective, you set the standards. What's the minimum standard that's acceptable for that objective? Based on the objectives, you choose parents and you make crosses in pairs. That gives you seedlings. And first, you evaluate the original seedlings. Each seedling is different. The two parents are highly heterozygous clones. Every seedling from that cross is genetically different, and the seedlings from the different crosses are different from each other. So we evaluate in two phases. We evaluate the original seedlings, identify the best, propagate them, and establish replicated yield trials. The trials give us the data to justify release of a new cultivar. There are two major categories. The first is suitability to the blanched kernel market. That is, we want white kernels with the brown skin, the pellicle removed. Our major buyers are chocolate makers and bakers. In the field, we want bud mite resistance. That's an aerophyid mite, and there's a lot of genetic resistance. We don't want to have to spray for something if we can find genetic resistance to the pest. Round nut shape. Nobody wants long nuts, but most of the world's hazelnuts have long nuts. High percent kernel. That means thin shells. Precocity. We want our trees to start to produce nuts at an early age. Seedlings by the fifth leaf. In a grower orchard, we want to see a few nuts in the third leaf. High yield every year thereafter. Easy pellicle removal. We roast with dry heat and then rub the kernels to see if the skin comes off. We would like most of the skin to come off. Few defects. We want few defects in a future slide here. I will show you the different types of defects. So we take a sample typically of 100 nuts per tree, and if there's too many defects, we mark it for discard. Early maturity. We want our nuts to fall to the orchard floor earlier than Barcelona. Barcelona ripens the first week of October, and in Western Oregon, the fall rains begin first week in October. We would like to finish our harvest before the rains begin. The number 9 on the list, free-falling nuts. We use a lot of Turkish material. Turkish hazelnut cultivars have long clasping husks that hold the nuts on the tree at maturity. And we select in the offspring for free-falling nuts. The other major category is resistance to eastern filbert blight, and we use both major gene resistance and quantitative resistance. So this is the flow chart or the timeline from cross to release. We choose parents and make crosses at the beginning. We grow the seedlings in the greenhouse the first year. We grow the seedlings in the field. We evaluate them as soon as they start to produce nuts. And by the third or fourth crop of producing nuts, we've whittled the number down substantially to less than 1%. We propagate the selections by layerage. We line the layers out in a nursery row for 1 year and then plant replicated yield trials. We collect data from those trials in years 3, 4, 5, 6, and 7, and with the data, decide which, if any, are worthy of release as a new cultivar. Okay, 17 years from cross to release, but don't be misled. As soon as something looks better than its parents, it's immediately used as a parent in further crosses. So from seed to seed, the timeline is 8 years per generation. These are some pictures of the breeding process. Upper left, those are our pollination cages. The trees inside have been emasculated by clipping off the catkins, covered with white polyethylene. There's a wooden frame to hold the polyethylene in place over the tree, and there's a bit of bird netting at top because sometimes bird likes to land and poke holes in our white polyethylene. We collect catkins when they are newly elongated, let them dry in the lab overnight, and then pick up the catkins, funnel the pollen into a glass vial, stopper it with cotton, and place it in the freezer. Since one catkin can produce 4 million pollen grains, collecting pollen is not usually a problem. That's done in January, usually the first 3 weeks of January. And then the females become receptive. And then we take the vials of pollen out to the farm in a little ice chest, enter the cage in one corner, apply the pollen by fingertip, count the number of flowers as we go, write it on the tag, and then exit, nailing the cage shut. Now, we typically make 50 crosses per year and apply 2 pollens per tree. Before we start to apply the pollen, we put another sheet of white polyethylene through the tree to separate the 2 halves of the tree. We apply 1 pollen to one side and the other pollen to the other half of the tree. In late August, the hybrid seeds are ready to pick. They're allowed to dry down for a few weeks, and then they're soaked in water, and the stratification process begins. They're stratified in moist vermiculite, sometimes sphagnum moss, sometimes peat moss, at cool temperatures for 3 months. When we see root tips emerge, they're ready to plant in the greenhouse. We plant only those that have root tips showing. The others we put back into stratification for another 2 or 3 weeks. Then we pull the seeds back out and plant some more that show root tips. So germination is staggered over several weeks, but that's a natural survival mechanism in nature. It's a pain to deal with in breeding, but that's what we do. Okay, so producing hybrid seeds is fairly easy. You can produce thousands of seeds without much trouble. Much easier than walnut, for example. We grow the seedlings in the greenhouse in the first summer. We start out in small pots and then transfer them to 1-gallon pots. If we were to plant the small seedlings in the field in the spring, they might be 6 inches tall by the fall. If we plant them in the greenhouse and grow them for the summer, they're 6 feet tall by the end of the summer. So you essentially save 1 year on the breeding cycle by using the greenhouse in the summer. In early September, we take them out to the farm, and in October, we plant them in the field. 3 feet apart in rows 10 feet apart. Typically 5,000 seedlings per year. That's 50 crosses times 100 seedlings per cross. Okay, evaluation. Seedlings produce a few nuts in the 4th year. Most of them produce nuts in the 5th year. If they don't produce enough nuts by the 5th year, they're cut down and we don't wait. So we harvest the nuts, we dry them, store them, and then because hazelnuts start to flower in December, the month of November is the 1 month between harvest and having the crossing plan in place. So time is of the essence. From each tree that we harvest, we crack out 1 or 2 50-nut samples and look for these different types of blanks that are illustrated. Different types of defects. Blanks, that's a shell with no kernel. Moldy kernels, shriveled kernels, poorly filled nuts. That is, the kernel only takes up part of the space inside the shell. Twins, that is 2 kernels in one shell. And the last category are the black tips, where the suture opens and allows the kernel to be exposed to air. Upper left is the blanching process, the rating scale from 1, which is complete pellicle removal, nice bright white kernel, over to 7, which is no pellicle removal, just brown kernels. And we would like ratings of 1, 2, or 3 for the blanched kernel market. Now, that's me on the left, cracking the 50-nut samples to determine the defect counts. We hire student workers to do the 10-nut samples. That gives us the percent kernel. And Dave Smith, for decades, did the evaluation, the roasting, the rubbing, and the blanch ratings. Evaluation starts in late August in the field. So I typically take one of my graduate students or another research assistant or worker, and we go row by row, tree by tree, inspecting the nuts and the bushes. We take notes on size, shape, crop load, and so on, mostly looking for reasons to discard trees. Of those that we see, we mark for harvest only about 10%. The other 90% are marked for discard. And it's hard to do that when the leaves are on, so most of the removal is in the winter months after the leaves have fallen off. Lower left, you can see beaver orange, OSU beavers. The colors are orange and black, so orange is a good color. Blue is bad. So the blue trees get cut by the chainsaw, the orange trees stay to live for another year of evaluation. Upper right, you can see the field after the chainsaw has been through. And lower right, you can see a field of seedlings where 5,000 has become 50. The numbers go down fairly quickly after the trees produce nuts. Okay, hazelnuts naturally produce suckers from the base every year. And we use those suckers to propagate our best selections. Uh, in June, we removed the smallest suckers, and from the larger suckers, we've removed the lower leaves. We place a metal ring around the base of the sucker. We spray on the suckers, the base of the suckers, IBA to induce rooting. We then enclose the suckers in a tar paper ring and cover the base of the suckers with moist sawdust. Then in December, we harvest the layers. And what we want to see is shown in the lower left. You have a sucker with a ball of roots at the base. That's right above where the metal ring girdled the sucker. Those rooted layers go into the nursery for 1 year and then are used to plant replicated yield trials. Our yield trials, that's me years ago. Our yield trials are typically 4 reps in one trial and another 7 in a second trial. With 11 trees, that's really the minimum to give us the data we need to justify release. Well, we have released 28 cultivars to date, the first being Willamette in 1990 and the most recent being Thompson in 2023. 13 cultivars, also 12 pollinizers. Since hazelnuts are self-incompatible, if you plant an orchard, you've got to be sure that there's compatible pollen in the orchard at the right time, that females on the main cultivar are receptive. Commercially important today, Theta, York, and Felix are very widely planted. We also released 3 ornamental cultivars with red leaves. So I'm showing trees and nuts of 3 of these cultivars to give you a feel for what we've released. This is Jefferson, the nuts and the kernels on the left, Polly Owen from the Oregon Hazelnut Industry Office in a Jefferson orchard near Mount Angel. Jefferson was released as a replacement for Barcelona. It has the dominant allele for EFB resistance from Gassaway, but has the size and shape similar to Barcelona, a nice upright tree that's easy to manage in grower orchards. Unfortunately, it's not earlier to mature. The nuts drop with Barcelona or up to 3 days later. They are 45% kernel by weight, which is better than Barcelona, which is 43. They are large nuts and quite attractive. Because the nut yields per tree are higher than Barcelona, but the tree is only 70% of Barcelona, the yield efficiency is considerably higher. Our growers like the growth habit of Jefferson because it's so easy to manage. One of our kernel varieties is McDonald. It's now one of the most widely planted in the Willamette Valley. It was released for the blanched kernel market. It has a moderately vigorous tree, high yields. The nuts are down on the ground 14 days before Barcelona. Excellent quality, thin shells, few defects, and it is cross-compatible with 2 other kernel cultivars, Webster and Polly-O. So you can plant 2 cultivars or 3 cultivars side by side, and they can serve as pollinizers for each other. Polly-O is the other one that's very widely planted in Oregon now, also for the blanched kernel market. It's a vigorous tree it's precocious and high yielding. The nut size is small to medium, round in shape, very good blanching, very good flavor. 14 days before Barcelona again, so a striking improvement in maturity date. It is cross-compatible with Webster and McDonald. Another release, not by OSU, although it's an OSU selection, OSU 541-147 was released under the name The Beast, trademark name. For the Eastern United States, where eastern filbert blight is such a serious problem. We don't recommend it for Oregon because we have better releases for Oregon, but this combines genes 1/4 from the American hazelnut and 3/4 from the European hazelnut. It has resistance from Corylus americana rush from Pennsylvania, and it is still resistant in New Jersey, where gas array resistance is no longer effective. You can see the nuts and kernels in the center right. Um, Not good enough compared to our cultivars, but better than anything that uh, is available in the East up until that time. There are new selections coming out of Rutgers. There are also materials from the Upper Midwest program, but the Beast was released to fill a need in the meantime and still looks good. This tree on the left at Rutgers, that's Tom Molnar and me, and that was the 19th year. It's been a consistent producer every year in central New Jersey. Qualitative traits in hazelnut. I'm a plant breeder. I get excited about genetic diversity in plants, and there is so much genetic diversity in hazelnut. And 4 of these traits gave me nice short publications so that I could continue to work as a professor on the faculty here. Contorted growth habit. The original selected in a hedgerow in England, sold sometimes under the name Harry Lauder's walking stick. This is a single recessive gene. Upper right, Barcelona carries a deleterious recessive, a chlorophyll deficiency. So if you cross 2 heterozygotes, you get 25% of the seedlings with yellow leaves. That's from our major cultivar. Bottom left, normal female flower with red styles compared to a female with yellow styles. The yellow comes from our leading pollinizer, Daviana, single recessive gene. And the lower right, the cutleaf hazelnut, I think was selected in France over 100 years ago. It has this strange leaf shape, sometimes called oak leaf, although it doesn't look like an oak leaf to me. So you've got cutleaf, and a normal roundish leaf on the bottom, another single recessive gene for leaf shape. But most traits that we work with are quantitative. Eastern filbert blight takes up a lot of my time, has a major resistance is a major objective of the program. This is our neighbor's orchard next to our research farm. Anise is a highly susceptible cultivar. It essentially melts when it's exposed to eastern filbert blight, and the trees succumb very quickly. First, you lose a few branches at the top, then you lose the major scaffold limbs, and eventually the entire tree will die. This is a native fungus, native on Corylus americana in the eastern half of North America. We grow the European hazelnut in Oregon. Most European cultivars are highly susceptible, but sometime around 1960, somebody introduced infected plants into Southwest Washington, and the fungus spread throughout the Willamette Valley over the next 30 years. The fungus has a 2-year life cycle. You don't see any symptoms of disease in the first year after inoculation. You have to wait about 16 months, 18 months to see any sign of cankers. So it's a difficult problem to work with. But fortunately, over time, with all the diversity that we've brought in, we have more than 100 sources of high-level resistance. That is, no disease in our tests. And of those, it says 35 have been mapped, and that was the case during the meeting in Missouri. So we have used DNA markers and our reference linkage map to place these EFB resistance genes on chromosomes. Which chromosome or which linkage group, and what is the location? Well, these on this slide all map to the same location as gas away resistance, the original source that we mapped. So you see there's trees from Minnesota, Spain, Russia, Serbia, southern Russia, Crimea, Corylus heterophylla from South Korea, another interspecific hybrid from Cecil in Michigan. All of these resistances have mapped to the same position on linkage group 6. Another region is on linkage group 2, the first being one from the Republic of Georgia, followed by a couple from Holmsky, Russia, that's near Krasnodar down in southern Russia. Turkish material, the resistance is mapped to linkage group 2, and also a selection from seeds brought in from Sochi, Russia. But Sochi is right next to Abkhazia, which used to be part of Georgia. So it's probably the same as the Georgian source. But a lot of different places that we picked up resistance that maps to linkage group 2. The third major location where resistance is mapped is on linkage group 7, the first being Ratholi from Spain, followed by one from Sochi, Russia. I mentioned the beast and its resistance from Corylus americana and Rush. That maps to linkage group 7. Yoder number 5 from Russell Yoder, Northern Nut Growers member from Smithville, Ohio. Two selections that we received as science from Moscow, Russia, and two seedlings from seeds collected in Crimea. All of those map to linkage group 7. This gives us a wonderful diversity of resistance to work with in breeding. Okay, I mentioned that most of the traits that we work with are qualitative, or a few are qualitative, most are quantitative. That is, you can measure the traits and determine how much of that is passed on to the offspring versus how much is just due to non-genetic factors. I was challenged early in my career by my predecessor, Maxine Thompson, who said, "I don't think those defects are heritable." And I took that as a challenge to show her that indeed they were heritable. And so for the first few years, I cracked nuts and then did a study regressing progeny means on mid-parent values and showed that indeed many of these traits are quite highly heritable. Defects are passed on from parent to offspring. Overall, good kernels, 42% heritability. Heritability ranges from 0, not heritable at all, to 100%, complete genetic control of the trait. So a breeder, if they see anything over 40%, is thrilled that it's a highly heritable trait. Doubles, 2 kernels in one shell, highly heritable. Moldy kernels, highly heritable. Kernels with black tips, highly heritable. Blanks, still highly heritable, but a lot of variation around the mean. Poorly filled nuts and shriveled kernels, not so heritable. Probably a function of crop load. If you have too many nuts on a small plant, the kernels don't look good after you harvest them. And brown stain that we now believe is due to stink bug feeding, not very heritable. Some more quantitative traits. If you look at nuts, the length, the width, the depth, any measure of shape, they're all highly heritable. You only need to look at one nut to know if it's round or not. You only need to look at one nut to know if it's a big nut or not. Nut weight, another measure of size. Kernel weight, highly heritable. Percent kernel, how thick is the shell? Very highly heritable, and it's a cultivar characteristic. Amount of fiber on the pellicle, another cultivar characteristic, although there's some variation from year to year. And blanching, how much of the skin comes off the kernel when it's roasted? Highly heritable trait. So I am thrilled as a hazelnut breeder to have so many of my important traits be highly heritable. Husk length. Let's look at the middle number, relative husk length. That is husk length relative to nut length. 91% heritable. So I can work with Turkish material and select for free husking types fairly easily. And I only need to look at one nut cluster to know if it has a long husk or a short husk. Another set of traits are the phenology traits. When do the stigmas come out and are receptive to pollen? When do the catkins elongate and shed pollen? When do the leaf buds break in the spring and shoot tips emerge? And nut maturity in the fall. When do the nuts drop on the ground? These are all highly heritable traits you can select based on performance of a single plant quite accurately. And to remind you, if you're not familiar with hazelnuts, they are monoecious. They have separate females. That's the arrow pointing at the top. Each female has potential to develop into a nut cluster. The male inflorescences are catkins. And as I mentioned before, one catkin has the potential to shed 4 million pollen grains. And they flower for us in midwinter. Mid-January is the peak of pollen collection for us. Okay, another really important objective is resistance to eastern filbert blight. I showed you the major genes and where they map to. We also work with quantitative resistance, that is fewer cankers and smaller cankers. And we do this by exposing potted trees under a structure and putting diseased branches on the top of the structure, turning on the sprinkler irrigation periodically using a timer. When the branches, the inoculum gets wet, they exude spores. The spores rain down on the foliage on the young trees below. And then 18 months later, you can see some cankers develop. Well, we've done this with hundreds of selections and have found over 70 that have good quantitative resistance and also good nuts. Our two Czechs are Tona di Giffoni from southern India, southern Italy, and Sacagawea from our program. Those are standards. We would like quantitative resistance to be equal or better than those two Czechs. I will mention that Rutgers University has released 3 cultivars with quantitative resistance, Raritan, Monmouth, and Hunterton. And they have in their pedigrees Sacagawea and Tona di Giffoni. Okay, now at this point, I'm going to try to expand my presentation to things that are applicable to other temperate tree nuts, not just hazelnut, but hazelnut, I think, has a lot to teach others. Plant breeding is a numbers game. Doesn't matter what crop plant you work on, you've got to grow thousands of plants to find the very few that are worthy of release as new cultivars. And we're dealing with clonal cultivars. So you find one good plant, you clonally propagate it, and that becomes your new cultivar. Breeders of all of these plants decide the number of crosses per year and the number of seedlings from each cross to plant. Breeders tend to maximize the genetic diversity in what they plant. So as many crosses as possible and a representative number of seedlings from each. My choice is 50 crosses per year, 100 seedlings per cross. So that's 5,000 seedlings every year. The odds of success, that is finding something worthy of release as a new cultivar, improves when you work with high numbers. So if your program works with 100 seedlings per year total, you may spend a whole career before you find one worthy of release. I do have, sorry to criticize colleagues, but I do have colleagues overseas who have made crosses for 1 year or 2 years and then evaluated the seedlings for the next 30 years. And that's not a breeding program. You've got to move through the generations quickly. You've got to make crosses every year. And you've got to be ruthless in eliminating plants that don't meet your objectives. I've also seen colleagues in many countries that collect cultivars, and they evaluate the cultivars they've collected and publish data often. But if you don't make crosses, that's not a breeding program. That's a cultivar collection. I teach, have taught plant breeding every winter since 1989. In spring term, more recently, I also teach a second class in breeding clonal crops. And one of the topics is breeding strategy. And I like this chapter in Methods in Fruit Breeding published, and the chapter's by Royce Bringhurst, published in 1983, quite a while ago. So if you go to your supermarket today and can find fresh strawberries for sale, you can thank Royce Bringhurst to develop the first day-neutral strawberry cultivars. Very productive strawberry breeder at the University of California, Davis. And in his summary chapter, which is short and sweet and very much to the point, he makes several points that I would like to share with you today. First, he says long-term financial support is needed for applied breeding from government agencies and from the commodity groups that the programs serve. If you're not willing to commit long-term financial support, the program will not succeed. It takes time. The program must have clearly defined objectives. That is, for each objective, there must be a minimum value. Clearly defined priorities, procedures, and standards. You've got to have a clear picture in your mind what constitutes a new and better variety. Next on the list, the breeder should start at the most advanced level. Do not reinvent the wheel. If you've got something with the trait that's almost ready to release, use that as the parent. Don't go back to the beginning. Eliminate seedlings and selections that fail to meet the objectives. In other words, eliminate the losers and move through the generations with dispatch. A breeding program should have breadth. We talk about one breeding program, but in fact, breeding programs have several different parts, and each part moving forward with different objectives. And in order to be successful new cultivars, we have to know how to manage them and what are the appropriate cultural practices, management practices for them, including what's the appropriate spacing between trees in the orchard. What about other tree nuts? Talking about almonds, Persian walnuts, and black walnuts, pecans, and so on. Plant breeding requires a long-term effort and financial support. And in today's world, that's hard to come by. Federal formula funds, that is Hatch Act funds, and state funds appropriated by the state legislature are limited. And agriculture in many states is not a priority. Grants, there is grant money available, but they are highly competitive, and the typical grant lasts for 3 or 4 years, not the 17 years it takes from cross to release. So several commodity groups provide direct financial support. And not only do they provide support from their growers by taxing their growers, but they also send people to the state capitol to lobby the state legislature. They also send people to Washington, D.C. to lobby our politicians there. Say, we growers are putting in this much money. We would like the state government to match it. We would like the federal government to match it and share the burden. So in Oregon, we have the Oregon Hazelnut Commission that supports research. In California, they have similar organizations that support breeding in almond, walnut, and pistachio. The chestnut is a different situation. People trying to restore the American chestnut do have a long list of donors, and they've kept money coming in for those efforts for many decades now. Different source, but it takes money to keep these efforts going. Okay, diverse germplasm is the foundation for genetic improvement. And in all of these temperate tree nuts that we just looked at, we have a lot of genetic diversity available to us. The USDA ARS maintains collections of the major nut crops in various locations. Corvallis for hazelnut, Davis for Persian walnut, black walnut, almond, and pistachio, and College Station, Texas for pecan. The species native to the US include pecan in the southern states, eastern black walnut, and American hazelnut, also the beaked hazelnut and the California hazelnut. The American chestnut, native to eastern North America, still persists as stump sprouts throughout its range. So we haven't lost that genetic diversity. We hardly ever see nuts produced. The Persian walnut, there are types for cool or cold climates that exist in Ukraine and the Carpathian Mountains of Poland. It is now more difficult to collect plant germplasm in other countries We now have an international treaty that recognizes that countries own the genetic diversity within their borders, and we must negotiate with the governments to have permission to bring material out of the country. So the US Senate approved that treaty 5 or 6 years ago. So after about 20 years of delay, the US is now a signatory on that treaty, and that regulates what we are allowed to do as collectors. Okay, to remind you of almonds, it's grown in California in the US, big production in Spain also. Almond is closely related to peach. If you look at the plants, you look at the flowers, you look at the fruit, you say, well, that's a peach, except we eat the seeds inside the shell. Breeding objectives in recent years have been for self-fertility because the traditional cultivars are all self-incompatible. And late blooming. Standard cultivars flower very, very early in the spring. Spring frost can easily knock out your whole crop, which is a big limiting factor. But now there are types that flower 2, sometimes even 3 weeks later than the standard cultivars, and breeders have made tremendous progress in both of those areas. Persian walnut, and for some members of the Northern Nut Growers Association, English walnut, but there's none of these native to England. So let's use the more correct term. The Persian walnut. Chandler from UC Davis is the preferred cultivar, is lateral bearing. It has thin shells. It has beautiful creamy white pellicle, good flavor, and it's planted all around the world. And the bottom pictures show an irrigated orchard and the lateral bearing habit of Chandler. Juglans species, the walnut species, there are several that are native to North America. And the important ones, Juglans nigra, the eastern black walnut, is from eastern Kansas to the Atlantic seaboard and up even into southern Ontario, Canada. The butternut is another walnut species. Its distribution is just a little bit further north than eastern black walnut. There are several minor species. Juglans heinzii. You see the red arrow over there in California pointing to Juglans heinzii. That's Davis, California. The species has a very narrow range for its distribution, but a huge impact because it's been used as a rootstock in Persian walnut orchards in California, and more recently, hybrids between heinzii and the Persian walnut are clonal rootstocks. So we have a lot of genetic diversity in Juglans, and you can cross those species with each other. I took this picture when I was visiting Tom Olner at Rutgers. This was June of 2016 to show you the size of Juglans nigra in central New Jersey. And that's Tom standing there by the fence. And you can see how big that tree is. Juglans nigra is very important for wood production, for furniture and paneling. The nuts have very thick shells and a very strong, typical aroma. And there are efforts to further domesticate eastern black walnut. There is a breeding program now at the University of Missouri to improve eastern black walnut for nut production, not just wood production as we have had for hardwood trees at West Lafayette, Indiana for several years. This picture was also taken in 2016. That's Mike Gold at the University of Missouri Agroforestry Center. We visited here in 2023. Yeah. We have some black walnuts here in the Willamette Valley of Oregon. And on the left, you can see a big tree down by the Willamette River, and that's me standing at the base. And people who buy hardwood tell me that many of the trees in Oregon are actually hybrids between Juglans nigra and Juglans heinzii, and they show the hybrid vigor of this interspecific hybrid. But anyway, this tree is huge, absolutely huge, but it sits only 100 feet from the Willamette River, so there's no shortage of water reaching its root system. I mentioned Juglans heinze and Juglans regia cross with each other and have given rise to the series of rootstocks called paradox hybrids. So these black walnuts have many uses in commercial production. Okay, what about the other tree nuts? How similar are they? You may go to the supermarket and not see a similarity, but if you look at the botanical classifications and you look at other recent studies, you'll see that hazelnut, chestnut, walnut, and pecan are all members of the taxonomic order Phagales. That is, they are closely related, although they don't look that much like each other. According to Nikolai Vavilov, Russian scientist who established the best collections of plants in the world during the 20s and 30s, his law of parallel variation predicts that if you see variation in one species, you expect similar variation in related species. So that means in our context that variation in hazelnut you would expect to see in all of these other temperate tree nuts if you look hard enough. Interspecific hybridization has been successful in really all of these temperate tree nuts that we've talked about today. In hazelnut, Corylus avellana, the European hazelnut, hybridizes readily with Corylus americana, the American hazelnut, and Corylus heterophylla from Eastern Asia. Those crosses work in all directions very well. Other Corylus species are further removed and more difficult to cross with. Okay, how closely related? The Joint Genome Institute is a function of USDA partnering with the Department of Energy. And they set up this program to sequence the genomes of many plants that cover the diversity of plant species. And in this picture, this is more than a decade old, but this is the range of species that they chose to sequence the genomes of. And if you look at number 1, 2, 3, 4, 5, 6, and 7, you see cucumber and peach, Cucumis sativus and Prunus persica. Now, I would not look at those plants and say hazelnut is closely related to cucumber and peach, but it is. If you look at the genome sequence, they're the ones that are most similar. So the other ones that are kind of nearby are Medicago truncatula, that's black medic, and glycine max, that's soybean. So this is useful, and the monocots are down at the bottom of this figure. So hazelnut is related to cucumber and peach, but it's more closely related to those other temperate tree nuts. So if we look at this picture, it's actually a composite of two. On the left, you see the order Phagales. Hazelnut is included in the order Phagales, right next to the cucurbits. And if you further expand the order Phagales, you see the families, Fagaceae, that includes chestnut and beech. You see Betulaceae that includes hazelnut, birch, and alder. You see Juglandaceae that includes walnut and pecan. So these are actually fairly closely related. And my point is that breeders need to work together. The more closely related the species, the more useful is the information from one to the other. Okay. And that figure came from the Tree of Life web, Toll web, so that you can find that online. What about the other tree nuts? For those values that are published, the heritability estimates for the important traits are generally high. That is, you can select based on phenotype, and you don't need a big sample size to judge the phenotype. That's really useful. Today, genome sequences allow rapid development of new DNA markers, whether they be SSRs, simple sequence repeats, or SNPs, single nucleotide polymorphisms, or other types of markers. If you have a series of genome sequences, you can develop markers fairly quickly. DNA marker studies have identified major genes in hazelnut for eastern filbert blight. I showed you figures of where resistance has mapped to. In Persian walnut, there's a lot of good mapping work that's gone on at UC Davis. Patrick Brown leads the program there today. Lateral bearing, a very important trait. Chandler is the leading cultivar because of its lateral bearing and high quality. That is conferred by a major locus on chromosome 11. And if you believe Vavilov's theories, and there's a whole lot of data to support them, we should be able to map lateral fruiting in eastern black walnut too. And I would not be surprised if that came out to be one major locus in the same location. When we hosted our Hazelnut Congress in Corvallis in 2022, my assistant, Dave Smith, gave a presentation about the history of the program and our thoughts on why we have been successful. And I'll share these with you. It may sound like we're boasting, but we do have a lot to be proud of. We have had, since the beginning, a dedicated team, a project leader, research assistants with very little turnover, graduate students who come in and get their degrees and move on to better things. We've hired a lot of undergraduates and other seasonal employees, and we have research farm staff that is long-term. So that consistency over time has really helped us do a lot. The research farm is only 10 minutes from the university campus. Students can ride their bicycles out to the farm to work. I can go to the field and collect flowers and come back to the lab in a fairly short time. I know that at other universities, the research farms are hours away from campus. So we have a real advantage there. Our greenhouses are right on campus. I can walk from my lab to the greenhouse. We in the United States have our land grant university system. So every one of the 50 states has its land grant university established in 1862 to teach agriculture. And since that time, the teaching came first, then the research was added to it some 30 years later, and then the extension back in the 19 teens. So our land grant universities combined teaching, research, and extension. And we serve our stakeholders. We regularly communicate with, in my case, Oregon's hazelnut growers, and we conduct educational programs to serve their needs. We listen to their needs and respond to them. I grew up in Western New York. Cornell University was the land grant university. And I grew up on a family farm where we produced certified seed, and we delivered seed to the seed lab in Geneva, New York, which is part of Cornell University. So I grew up as part of the land grant system, thinking that this is normal everywhere in the world. It's not. We in the United States are so fortunate to have the land grant university system for teaching and for research and for extension. This transfer of new knowledge from the university to growers and the technology that goes with it has allowed the quick adoption of new cultivars from our program. So we can release something, get it into micropropagation before release, and have trees available in large quantities at the time of release. That's the theory anyway. We don't always make the time frame that we have in mind, but we have had clearly a major impact on Oregon agriculture through work here at OSU. We have a group of stakeholders that are tremendous people to work with. We are so fortunate. And in many families, I know 4 generations in the same family. We have the Oregon Hazelnut Commission, which is officially a branch of state government, but it's an organization that allows growers to tax themselves. The tax is collected by the handlers at the point of delivery of the nuts, and that goes into the research fund and the Hazelnut Commission decides which research projects get how much money. Money and equipment are sometimes donated by individuals in the industry or by processors in the industry. We've been fortunate to have received 3 different USDA competitive grants, although those are short term, and there's a lot of years with little money in between. We have an ongoing agreement with USDA to fund some of the EFB research. It funds about a half-time technician. We also receive extra money from the Oregon State Legislature for the breeding program. Again, that's enough for about a half-time technician. And in recent years, we've been able to create a royalty stream that provides new money to continue the work. So we have, since 2010, been patenting our new cultivars with US plant patents and also licensing them in Chile starting in 2010 and more recently in Europe, allowing us to recapture some of the cost of investment from people who did not pay into the system that developed them. So that's been another source of money, very important for us. So tree nut breeding, if we look at all of these temperate tree nuts, and the northern nut growers are excited about many different temperate tree nuts, there are large amounts of genetic variation for all of them. The status of the collections around the country varies. There are many instances where additional collection and preservation efforts are needed. Many important traits are highly heritable. Phenotypic selection works very well, and it's straightforward. Breeding requires long-term financial support. Breeding requires committed personnel who are going to stick around for decades. Stakeholders do give money that support breeding programs in 4 of these temperate tree nuts, 3 in California, 1 in Oregon. Pecans struggle. Chestnuts, their work depends on donations from individuals. Competitive grants, specific cooperative agreements, state and federal earmarks, and royalty income are other ways that breeding programs are funded. And it really takes a combination of all of the above to keep a program going.
Okay, that is the end of the recording there. So, Sean, if you'd be willing to come back on, let's get into some questions. If folks have questions, they can certainly type them into the chat. Also, if you want to raise your hand, you can ask, I guess, directly. We have a small enough group here that that works as well. And to get things started, Sean, I'm curious. Early in your presentation, you listed off a bunch of new places that were getting involved with growing hazelnuts. And I'm curious, is there any sort of a limiting factor to who can get into the game, I guess, to grow hazelnuts?
Climate is the major limitation. In North America, we have eastern filbert blight to contend with, so that's an additional hurdle. And the fungus that causes eastern filbert blight is native on Corylus americana in the eastern half of the United States. But climate is a limitation everywhere. We do have a very good situation here in the Willamette Valley. We're 1 hour from the Pacific Ocean, so it's never too hot, never too cold. And the trees flower in wintertime when it's very humid. So it's like a refrigerator for a few months. We always have good overlap between pollen shed and female receptivity. So we can count on a crop of nuts every year. In other places, particularly where you have those huge fluctuations in temperature between day and night and 1 week and the next, you can have pollen flying a month before the females come out. Cold hardiness you need in northern zones. I cannot evaluate cold hardiness here. We have temperatures that are more like North Carolina. So those are the two major factors.
Any other questions for the chat or chime in? Okay, Sean, real quick. So you said that you were growing up in Cornell, so you were out east, and then I spent a little time at Rutgers. How in the world did you get pulled to the other side of the United States and end up in Oregon?
I was offered a good job. And I
I
do have
suppose.
some family in Washington State, so I'd been out west before.
Okay.
Sean, if I may, it's Jerry Hendon from New York.
Hi, Jerry.
I've worked with Peter Harmon on
Yeah.
Aqueduct, Long Island, and last year he had a tremendous crop. This year, almost nothing, and he was surprised to find out that hazelnut, at least in his area, doesn't bear every year. Is there any advice that you could give him?
Well, it's like I tell the growers in California, I said, no, you can't grow hazelnuts there because they can grow everything else. Don't compete with us. Long Island, though, has a pretty mild climate. I mean, it sticks out into the Atlantic Ocean, and I mean, central New Jersey has a very mild climate too. I think the one secret there is you have to have a mixture of many different types to make sure that you have cross pollination. And
Hmm.
if you've only got one cultivar, one pollinizer, well, that worked in Oregon, but in most places that's too risky. And if you're in a colder climate, you can have the winter injury. The catkins are especially tender. If the catkins are young and tight, uh, they can tolerate fairly low temperatures. But once the catkins elongate, it doesn't take very cold to kill them. That's usually the challenge in slightly colder situations that the catkins freeze and there's no pollen when the females come out.
Okay, thank
So,
you.
Sean, question here in the chat. For seedlings from Rutgers, what is the range of time in the year when catkins come out for flowering?
Seedlings from Rutgers. You mean the cultivars they released?
Yeah, yeah, yeah.
The releases from Rutgers were from crosses made here at OSU, selected in New Jersey and released. So it's pretty much the same time they would flower. We don't yet have them back here because we'd have to bring them in through the state quarantine to prevent introduction of Eastern filbert blight. The timing would be much the same here. Pollen shed in the middle of January and female flowers receptive in the first half of February in our conditions. I'll also say, uh, since I have a few minutes and a fairly small audience, in our collection, it's an incredibly diverse collection. We have pollen that is pollen shed starts around Thanksgiving time, and we still have pollen being shed now up until about the 10th of March. We have females that become receptive in the middle of December. We still have females that are receptive now. So in a collection of thousands of different plants, we have no problem getting a crop of nuts every year. But it's hell for someone who has an allergy to hazelnut pollen.
I hear you.
Wait, Sean, do you have that? Are you allergic to hazelnut pollen?
Yes.
Are you just revealing this now?
No.
Okay.
I remember when we were at the Connecticut Experiment Station, we were growing up the collection of the USDA cultivars of corn. And the old corn breeder who came to help me with this in the 60s, the first thing he asked me was if I was allergic to corn pollen yet. And I said, no. He said, you will be.
And the Willamette Valley is the grass seed capital of the world. And we have many grass breeders out here, and most of them are allergic to grass pollen. It's the constant exposure year after year after year.
Hmm.
Sean, speaking of corn, Jim asked in the chat here, corn breeding and strategy, you only grow 100 seeds per cross. Why so few? What's the magic 100 number?
Yeah, I could work on Arabidopsis and grow a population in one flat in the growth chamber, but trees are bigger. And we pay for
Space.
the land we use $90,000 a year in land use charges, another $9,000 a year for the greenhouse space that we use. You have to fit it within your budget. I think 100 seeds is enough to get a good idea of the potential of the cross, and also good enough to identify the really good parents that you want to use again.
Great answer.
The computer said thank you
What?
in the chat. Yeah.
Hmm. That's probably Jim. He's
Yeah.
like a computer sometimes.
He's incognito.
Yeah.
Well, 100's a good number, Sean. A lot of groups have used less than 100.
Yeah.
At Davis, we would shoot. That was always our aim, but we never made it, or rarely
Right.
made it. Rarely made it.
It's
Sometimes
more
you get
challenging.
12.
Yeah,
Right.
it's more challenging because in walnut and also in pecan, I mean, first of all, they're huge trees, and you have to get up and emasculate, and you have to bag the individual flowers. We just emasculate the entire tree.
Yeah.
And we have typically 400 females on one half and another 400 females on the other half, and it's fairly easy to make the pollinations and harvest the seed. So
Yeah.
we're lucky in that regard.
There's
I,
no there's
I've
no
never
question.
been,
Sorry, Sandy. Go ahead, Sandy.
I've never been able to get 100 progeny to test, and Jean keeps telling me that's
Yeah.
not enough. I'm lucky if I get 25 planted out on our 75 acres. I keep taking over the farm.
So yeah, we all get accused of that.
Yeah.
So that's a reason, interesting, Sandy, that Jean Romero Severson, to create her mapping populations in oak, she chose two oak trees near each other on the Purdue University campus and harvested the open pollinated seed. And using the SSR markers, she identified the full SIB population. And that's how she achieved that population size. But by doing it by hand, it would have taken decades.
Mhm.
Fascinating.
Well, hey, I did wanted to thank you, Sean, because you did. This was a great talk. I sent it around to a few students and colleagues around town here at Purdue today. And yeah, Owen Stiles, I saw he joined. And yeah, you'll be proud to know we were collecting from a 15-year breeding screen of butternut. We were collecting graftwood of new selections to graft out and start to distribute around the region here in the central hardwood region. We got 14 clones of butternut, and then we actually selected almost 10 walnut as well, too, black walnut for timber.
Good. And how are you doing against the butternut canker?
Yeah, we're doing quite well, really. It's been interesting. The hybrids, absolutely. The disease comes from Asia, so the Asian Japanese walnut, heart nuts seem to be the host with resistance, right? Probably came in not unlike chestnut. And so we see that, and we've got a wide variety of back crosses. It's quite amazing. We've had a really good molecular guy, Aziz Ebrahimi, just recently do genotyping by sequencing on a vast number of them too. So yeah, we were getting a pretty impressive DNA profile now on them too. But this has all been through inoculation, creating epidemic conditions, and then time, 15 years. And that seems to be about right. It was interesting too, working with, with the students here, showing them previous selection marks and like, okay, is this one still in or is it out? And you see how that changes and falls apart over time too. So yeah, I'm pretty hopeful. We're going to have some pretty good stuff coming out. We found a little more resistance in native butternut, and we preserved some of those populations for people to grow. So yeah, the whole future of butternut right now is bright.
Good. Good to hear.
But nascent. I mean, I love the way you covered everything and talking about the structuring of the program and the funding and the staffing. I mean, you guys have it down very well, very impressive, and we should all be using you as a model.
Yeah. Time
And
to retire.
unfortunately, you can't really, well, you can retire, but you can't quit, right?
That's right. That's right. We never quit.
Not that,
Retire,
yes,
but we
Sandy,
never
back me
quit.
on this.
Well, let's, any last questions for Sean? Let's pick his brain while we're here. Otherwise, we can wrap things up.
I do have a
Sean.
question here.
Yeah.
So, hi, Sean. Great presentation.
Hi.
So you talk about the importance of having industry support and using that to fund the breeding. What do you do in an instance where the new variety is what you need to foster the industry? How do you find the synergy to build this? A lot of these kind of orphan crops don't have that support.
I'm thankful that our growers recognized the need some decades ago. It is a challenge. One of the things that helps us get research funding is that, well, in contrast, if you're doing apple work, the farmers would say, well, let the farmers in the other state pay for it, and we'll take the results. Oregon is 99% of the US hazelnut production. Our industry knows that if they don't fund the research, it's not going to get done anywhere. I think that's part of the package. So yeah, you're in the upper Midwest. It's a challenge because the number of acres is so limited and the markets are limited and the climate is a serious obstacle, and eastern filbert blight is present throughout the range. Yeah, it's a challenge. Do I have a
How
magic
did the
solution? No.
industry form in a way that growers group was formed? How did they enforce that everyone should buy into that?
Well, I think the details vary from state to state about what's possible. We do have a federal marketing order for hazelnut that gives good information on supply from Oregon orchards and supply around the world. It also gives information on the markets and what's open, what's potential. And that creates organization. We also have the Oregon Department of Agriculture, and all of these commodity commissions are officially part of state government. They are part of the Oregon Department of Agriculture. So there's government backing in the structure, and it takes a vote of that commodity group to set up a commission. And that allows that commodity group to tax itself for marketing purposes, more importantly for me, for research purposes. So it was an action by the growers in the industry that created the structure for both research and for marketing.
I guess I'm more wondering about essentially where the teeth are, right? So if someone doesn't want to pay this tax and they sell their nuts somewhere else, they would still benefit from a variety release, or is there some way for you to make sure that their varieties are only released to people who've paid in?
So two aspects, Adam. The funds for the research money for the commission is collected by the handlers. When farmers deliver their nuts, it's washed, cleaned, dried, and that they have the weight. And so there's automatically a record of how much was produced and sold. If you take your nuts to the local farmer's market or you sell nuts out of your garage, that's not in commercial channels. There's no assessment on those. But where are you going to sell the nuts from 100,000 acres. It's going to be through the handlers. And we have a system in
I
place
mean,
that
if you
allows them to quantify how much is delivered and to assess funds for research and assess funds for marketing based that
Any
I
other
answer a
questions?
little bit more. Another aspect is one reason that we are now protecting our releases with US plant patents and then seeking legal protection in other countries is that's a way to get those people to pay back for some of the research that benefits them, and they did not pay in advance. So that we deliberately keep the royalty per tree low. For Oregon growers, it's only 50 cents a tree because they have already paid through the commission. But this gives us a way to bring in money from people who did not pay to develop the cultivars. I think the royalty that Rutgers University tax on is considerably higher because they don't have a
so
grower organization.
it sounds like the key piece is logistically just having aggregation hubs that have signed on to this. And if it's more of a decentralized crop or crop biology, then it sounds like it'd be hard to enforce some sort of unified action like this.
Yeah, I think that's if I could give another example, for many decades, we had the Oregon processed vegetable industry to support vegetable breeding and other forms of research. But now there were only a handful of processors of vegetables in the state. So again, it was a fairly simple, relatively simple. But now another major group that's interested in vegetable varieties, new vegetable varieties, are the organic growers. But there's hundreds of those, and they're not organized, and there's not really a way to collect money from them to support research. Not an enforceable way. You can ask for donations.
It's past my bedtime, guys.
Ah,
Thanks, everyone.
Great.
Appreciate Sean.
it's
Thanks for
it's
joining
only
us.
5 o'clock here.